Hello everyone, welcome back to another video from shomus biology. In this lecture we are going to talk about amino acid titration math problems. ये biology math problems के series में हम अलग अलग प्रकार के problems को discuss कर रहे हैं और आज की ये video English and Hindi will be in the combination of both the languages and we are going to tackle the amino acid titration. Many students get puzzled with this, but believe me, if you watch this lecture, it's going to be super easy for you to solve this kind of problems. Although we'll solve two examples. For more examples, you need to get any of our service, but you'll get an overall idea कि कैसे solve करना है, आपको किस हिसाब से करना है, ठीक है? तो एक simple चीज आपको जानना है. You need to know only one simple thing. Okay? For all these problems that are there, amino acid titration, the problem is related to the three terms that is there. That is a pKa, the term is pH, the term is pi. You need to know the relationship between these three components, these three parameters, pKa, pH and pi. Okay? For easy understanding, we call pi as a principal investigator, the lab head. That's the short form of pi that we use. And in scientifically, the pi is the net charge. Pi is the pH where the net charge of the protein or peptide chain becomes 0. जिस pH पे एक प्रोटीन सीक्वेंस का, एक पेप्टाइड सीक्वेंस का ओवरऑल चार्ज 0 शून्य पाए जाते हैं, उसको pi कहा जाता है, जो कि ज्विटेरायोनिक फॉर्मेट में होता है, जहां पे प्लस जितना पॉजिटिव चार्ज एमिनो एसिड होंगे उतना ही नेगेटिव चार्ज एमिनो एसिड होंगे, तब जाके pi becomes 0. So that is something. Now, while pH and pK, what is the relationship between the two? आसानी से समझने के लिए pH को हम बोलेंगे phone, short form of phone, pH, phone, okay? And the relationship between pH and pKa is that for every single amino acid, let's say the amino acid itself is the alpha carbon, let's say, okay? There is a hydrogen attached to it, okay? And there is this NH, okay? So NH, right? R group, H group. This is something the structure looks like. तो इस हिसाब से अमीनो एसिड को देखने में होता है. So in this case what happen is that the amino acid we have a COO group here, इस side पे COO and this side NH. Now the NH can exist and it can be present in three different forms, NH3 plus, NH2, right? These are the two format the NH can be present. CO on the other hand can be present as COO minus, COOH. Try to understand this. I am giving you the most valuable information regarding the amino acid titration. For any kind of amino acid without considering the R group को consider नहीं कर रहे हम. Without considering the R group, अगर only amino group or acid group की बात करें, so for acid group there are two options, COO minus and COH. For NH group, amino group की बात करें तो वहां पे दो ऑप्शन है, NH2 और NH3 प्लस. NH3 प्लस मतलब यहाँ पे एक वन चार्ज मोर और NH2 मतलब 0 और COO माइनस मतलब एक चार्ज नेगेटिव, COH मतलब नेट चार्ज 0. That is the idea. For R group, there are different amino acids. For different amino acids, the R groups are different, right? There are charged R groups, there are neutral R groups, and there are so for charged R groups, there are positive charged R groups, there are negative charged R groups. Positive charged R group कहाँ पे आता है? Positive charged R group lysine, arginine, histidine. Negative charge R group DNE short form, that is aspartic acid, glutamic acid. Negatively charged R groups. So again for positively charged R groups, similar situation like amino group happens, NH3 plus और NH2 हो सकता है. For R और negatively charged R groups, aspartic acid, glutamic acid, वहां पे COO minus भी हो सकता है, COH भी हो सकता है. It will be CO minus or COH, right? That is something that you need to be very clear in your mind. This is the scenario of every single amino acid. Now the question they can ask you for a single amino acid, or they can ask a question of net charge of polypeptide chain where there are more than one amino acid. ऐसा क्वेश्चन दिया जाता है जहां पे सिर्फ एक अमीनो एसिड के ऊपर आपको निकालना है वैल्यूज, या फिर हो सकता है कि 3-4 अमीनो एसिड एक साथ जोड़ के बनाया गया एक पॉलीपेप्टाइड चेन, उसका वैल्यू आपको निकालना है. सो इन बोथ द केस यू नीड टू इंप्लाई द सेम नॉलेज, राइट? What is the protonation value for amino group? NH3 plus maximum, minimum NH2. For CO group maximum minus charge होता है, CO minus and COH हो तो न्यूट्रल जीरो होता है. So that is the idea. तो अगर एक amino acid की बात करें जहां पे in a single amino acid you have a CO minus and you have a NH3 plus, then what will be the net charge of this amino acid? So minus 1 here, plus 1 here, the net charge will be 0. Isn't it? This form, the pH, when the net charge of the amino acid becomes 0 is known as the Pi or isoelectric point of that amino acid. वो है Pi. Clear? So I am not only explaining the math, but I am also explaining the theory of Pi in here. Very, very important that you get to know about the theory very clear here. हो गया? Now once it is clear, let's look at the relationship between pKa and pH. मैंने कहा कि pH को याद रखने के लिए फोन टर्म को यूज करो, ठीक है? मैं यहां पे कहूंगा फोन रखो, हिंदी टर्म, in English it is drop the phone. Drop the phone, phone rakho. Why I said that? If you drop the phone, that means if you drop pH, that means if the pH is lower than the pKa, then what happens? Protonation happens. When the pH is lower than pKa, then protonation happens. pH वैल्यू जब भी pK से नीचे चला जाता है प्रोटोनेशन होता है. प्रोटोनेशन का मतलब है H प्लस बढ़ जाता है and the H plus will increase, so all the CO minus will be converted to COH, NH2 will be converted to NH3. जो COA minus फॉर्म में था वो COOH बन जाएगा, जो NH2 फॉर्मेट में था वो NH3 plus बन जाएगा. That is what the protonation does. That's the idea of protonation. That's protonation. What is deprotonation? The reversal of that. So COH থেকে CO minus बन गया और NH3 plus to NH2 बन जाता है. That is deprotonation. So reverse is deprotonation. So protonation कब होता है? When the pH is lower than pK, then protonation. When the pH is higher than pK, deprotonation. If you know all the things that I discussed here, the theory part that I discussed here, now you are ready to answer any question from the amino acid titration. And keep this in your mind. Phone rakho, put down the phone. Low pH, high pK, protonation. Low pH, high pK, protonation. The reversal is deprotonation. Clear? Good. If that is clear, I am going to erase some of the stuff here because I cannot work here without erasing it. You can take a screenshot if you like to, and I'll erase the data. Now I am going to solve the problem. अभी problem solve करके देखते हैं क्या है exactly. Okay. So now what is the pH at which the histidine amino acid, which has a pKa value 1.86 and 9.2. Now the pKa values are possible for all the different groups. What are the groups? CO, the carbonyl group, NH, the amino group, and the R group. Those can have different pKa values. So the pKa values, histidine is a positively charged amino acid. Positive charge R group is there. That's why histidine has 3 different pKa. If an amino acid don't have any charged R group, then there will be only 2 pKa. One is for the carbonyl group, another one is for the amino group. Okay. So here histidine has 3 pKa groups. So generally in exams the math questions will be asked from amino acids having 3 pKa values because it becomes a little more complicated to answer. So now at what pH the net charge of histidine would be 0. Okay. The net charge of histidine would be 0. Now try to understand, the net charge becomes 0 when there is Zwitter ionic form. That means CO minus NH3 plus. Then only the net charge becomes 0. We have talked about it earlier. हमने पहले बात किया कि CO माइनस भी रहना है, NH3 प्लस रहना है, एक -1 एक +1 देन 0 होता है. तो इसके लिए ना हिस्टिडीन की जर्नी जानना बहुत इंपॉर्टेंट होता है. So it's very important to know the journey of histidine, how it's going to look like. That is very, very important. Okay. So if we continue to increase with the pH value, so pH value कहाँ से increase करें? देखो यहाँ पे we have three criteria, 1.8 है, and there is 6.0 and there is 9.2. So we are going from low pH to high pH. Let me write low pH to high pH. Okay, moving from low to high pH in this direction. And we know lower the pH, more protonation, right? So more protonation would be in this side and deprotonation will be this side. Okay? So pH value that is lower than pK. This pK, so मान लो कि pH 1 पे क्या होगा? The lowest pH, all the pKs are more than pH 1. So in pH 1, all the three portions, all the three different groups, not portions, better to call it groups. Okay. So here this one is for the carbonyl group, this one is for the R side group, and this one is for the amino group, let's say. Okay. So at very lowest, the lowest pH, pH 1, what is going on in pH 1 is that the net charge क्या रहेगा? pH is less than pK, so all protonated. So the carbonyl group will be fully protonated here, COOH. Okay, carbonyl group fully protonated, amino group NH3 plus fully protonated, जितना protonation possible है. R group again fully protonated. So at very low pH like pH 1, the net charge here is +2. Okay. Now let's look at a pH which is more than this first pK value, that is the pH number, let's say 2. What's going on in pH 2? In pH 2 what will happen? So this carbonyl one will be deprotonated. Why? Because देखो pH 2 is greater than pK 1. pH 2, phone रखो, pH 2 should be lower to get protonated. So pH is more than pK, so it will be deprotonated. So this one will be deprotonated. Rest of the two will be fully protonated. So what will be the charge here? +1. Isn't it? Why? Because +2 minus 1, total charge is +1. So let me write it here, net charge. Now let's increase the pH more than 6. So we are looking at pH 7. What will happen in pH 7? What will happen? For carbonyl group pH 7, you see, pH is more than pK, so it will be deprotonated. For this amino group, what will happen? Amino group pK value is 9.2, which is more than pH. So for amino group, the pK is still higher than pH. So it will be protonated. It will remain protonated. But what happens to the R group? What happens to the R group here? R group's pK is 6.0, but the pH is 7. So the pH is more than pK, so it will be deprotonated. NH2 will be there. So what will be the net charge here? This is 0, this is +1, this is -1. So net charge 1 will be 0 in this case. Okay. And the last one, let's say pH 10. So now pH is more than all the pKs. Then what will happen to the C? Fully deprotonated. N group deprotonated. R group deprotonated. what will be the net charge? -1. So we are looking at the condition of histidine at varying pH. Lowest pH 1 to highest pH 10 in our example. Now for all this we need to take consideration for the 2 pKa values for which the zwitterionic form is presented. तो यहाँ पे सबसे महत्वपूर्ण चीज अभी यहाँ पे ना 3 pK वैल्यूज था, हमें कैलकुलेट करना था PI क्या है. The easy formula to calculate PI equals to pKa by pK 2 pK divided by 2. The average of 2 pKs, that's the answer to PI. But the biggest problem to answer this question was there are 3 pKs. So which 2 pK among the 3 we should choose? 3 pK दिया है, उसमें से 2 pK लेना है. कौन से 2 का एवरेज निकाले? वही 2 pK का एवरेज निकालना है जिस 2 pK के अंदर के फ्रेम में नेट चार्ज ऑफ एमिनो एसिड 0 हो रहा है. So if you look at the net charge of this amino acid, the net charge is 0 where? Here. So beyond that यहाँ पे +1 and after that -1 था. So the values that are floating, इसके पहले और बाद का जो pK का वैल्यू है, we need to use those two values. So the pK value that is before the net charge 0 is pK 6, after the net charge 0 is pK 9.2. So we must take these 2 pK and avoid pK 1.8. इसको अवॉइड करना है. क्यों अवॉइड किया हमने? क्योंकि जो नेट चार्ज 0 होता है उसके पहले का pK वैल्यू और अगला pK वैल्यू यही दो हम लेंगे. और यहाँ पे अगला पहला pK वैल्यू 6 था और अगला pK वैल्यू 9.2 था. सो डिवाइडेड बाय 2 दैट विल बी 15.2 डिवाइडेड बाय 2 दैट इज 7.6. सो pH वैल्यू होता है 7.6, आई मीन PI वैल्यू. तो यहाँ पे यही होता है एट 7.6 should be the correct answer. You can take a screenshot. बाद में बार बार वीडियोस देखना, तब इस स्क्रीनशॉट को स्टडी करना and you'll get to know about this important information is that for like it can be answered very fast. There are tricks to answer very fast, but I explained it sequentially because I think the theory is very very important for you to understand too. Theory भी उतना important है इसलिए उस हिसाब से समझाया. So you know that the pK values that we'll take floating the net charge of the amino acid 0. अगर यहाँ पे 0 आता तो ये वाला और ये वाला pK 1.8 and 6 लेता, right? And यहाँ पे आया तो 6 and 9.2 लिया है. So that will be the challenge for all these problems here, where they will generally ask you question regarding 3 separate pK and you need to get any 2 among them. This is the way to find it out. There is no shortcut. The more you practice, the better you would get to answer this kind of questions. Okay. Now this question will look much simpler to you now. ये बहुत आसान लगेगा इसीलिए क्योंकि थ्योरी पढ़ चुके हैं हमने. So the question stated which pH corresponds to the point at which lysine amino acid, the pK values given 3 pK values again for lysine, because lysine is another positively charged amino acid. It has a positively charged R group. So pK value 3 is fully protonated. In what pH corresponds to the protein point at which the lysine with 3 separate pKs is fully protonated? Fully protonated. अब देखो lysine को fully protonate होने में क्या करना है? If this is a lysine, then the lysine has 3 separate values. We have a C. Full protonation means carbonyl group COH form में रहेगा. The N group will be remaining as NH3 plus. And the R group will also be remaining as NH3 plus, right? This is the fully protonated form of lysine and the net charge should be +2 because this is 0, this is +1, this is +1. So net charge is +2. So this is the fully protonated version of lysine, and fully protonated version is only possible when the pH is lower than pKa. So to become fully protonated, the pH must be lower than all pKa's, all the pKa's that is out there. अगर pH की वैल्यू सारे जो pK हैं उससे नीचे है, phone rakho, remember, drop the phone. So pH value should be lower than all the pKs, then only all the separate groups will be protonated. So the pH must be lower than 10.53, lower than 8.95, lower than 2.18. So pH here, the value should be less than 2.18. So look at the option. pH 1 is lower than 2.18. So pH 1 should be the correct answer. It corresponds to the point where the lysine is completely protonated. pH 1 पर lysine completely protonated होगा. pH 2.18 पे क्या होगा? 2.18 तो मैच कर जा रहा है, तो नहीं होगा, 2.18 से नीचे होना है. And pH 1 is lower than 2.18. So that's why pH 1 is the correct answer, not pH 2.18. Right? And if they ask what is the pH where the lysine residue here, lysine amino acid here, will be completely deprotonated, तब क्या होगा? तब हाईएस्ट वैल्यू पे जाना है. मतलब दैट इन दैट केस द पीएच मस्ट बी ग्रेटर देन ऑल द पीके. सारे पीके के पार और ज्यादा होना है पीएच. तो यहाँ पे पीएच 11, पीएच 12, इस दोनों पीएच में ही डीप्रोटोनेटेड रहेगा. Lysine would be deprotonated in the pH 11 and pH 12, but it will be completely protonated at pH 1. Got it? Regarding the protonation and deprotonation. What is the relationship between pK and pH? pH should be lower than pK to be completely protonated. Phone rakho, drop the phone. Lower the pH, higher the pK, that is protonation. And lower pK, high pH, that is deprotonation. That is the funda to answer any kind of question from this amino acid titration as well as polypeptide charge distribution problems. ओके. तो चलिए अब अगर आपको कुछ और ऐसे प्रॉब्लम्स चाहिए, मतलब हो सकता है ना कि व्हेन यू आर प्रिपेयरिंग यू नीड सम मोर एग्जांपल ऑफ दिस कैटेगरी ऑफ प्रॉब्लम्स, उसके लिए ना बायोकेमिस्ट्री मैथ प्रॉब्लम्स यहाँ पे जाएंगे मेरे बुक के अंदर और इसमें आपको मिल जाएगा थोड़ा और ऐसे प्रॉब्लम्स. लाइक यू वांट दिस कैटेगरी ऑफ प्रॉब्लम्स, देयर आर सम पीएच प्रॉब्लम्स इन देयर, यू कैन सी some pH and acid based problems here in the page number 4, and also you will get the plenty of other problems. For example, the digestion problems are out there. Let me check it. There are some other problems related to amino acid titration, digestion problems are here. There are all digestion problems in the page number 19 in here. Protein purification and digestion problems, and you will also get half-life problems here in 23, page number 23. Half-life problems are here. Along with that, if you talk about the other problems, the enzyme problems, enzyme kinetics problems, detailed enzyme kinetic problems are here, right, in page number 31 or so. And Gibbs free energy problems are also there. Okay, now the page number might vary a little bit from edition to edition of our book because we are printing a new edition, so page number might vary, but the problems are always there. So you get plenty of, like, almost 5 to 10 problems in each category, and these problems are going to help you in order to understand, in order to answer the questions. So this is something that you practice. And last few CSANET question papers here is also mentioned, last few CSANET question papers, and these are all previous years biology math problems from CSANET life science examinations or uske answers. And not only the questions are there, but answer explanations are also mentioned. Answer explanations are also mentioned here in this section. Okay, so this category of problems you will get for this unit, that is biochemistry. You can check this out. You can get this book from us directly with a complete study material package of 3,900. Not this book, you'll also get 4 more books.